we should move to the last part of this first webinar, dedicated to pre-recorded sessions. So I hope that many participants were able to view your videos, so the two videos. But so I invite Andreas Holst and Debo Chakraborty to turn on their camera. And the way we can proceed, I don't know, is, well, it's we are experiencing this kind of interaction, so it's not really, we did not repeat it in advance. So what I suggest is to have 10 to 15 minutes per talk, I mean per talk, per presentation, per topic, and then even if maybe many participants were able to view your videos, I would suggest you, so it would be nice if you have a very short summary in 1 to 2 minutes, you know, of your work before starting the discussion of about 10 minutes per presentation. So let's start with Andreas, if you, so just very shortly, if you can summarize.
Sure. So my presentation was on the importance of host genetics in the transmission of infectious diseases. So a problem is, so my background is in animal breeding mainly, and now I'm doing a PhD in animal breeding and veterinary epidemiology at Wageningen. So currently, what we see in animal breeding is that infectious disease traits have a very low heritability, and a low heritability means that it is very difficult to select for such traits, and thus that the conclusion that you could draw from that is that genetics of the host are not very important in infectious disease transmission. So that's sort of the current belief. And what I did was try to find out whether that's true or not, because there are some limitation on the analysis that is done in general. So I did a simulation study and then try to estimate back the heritabilities and see what it actually meant in terms of the transmission. And then we find out that actually the low heritability is not a good indication at all for infectious disease trait, and that that's mainly because, of course, animals transmit the infection to each other, so they influence each other's disease status through their genetics, and that's ignored in the current analysis. And while the most interesting finding there was probably that it was in the simulation possible to eradicate the disease by genetically selecting the animals.
Thank you.
Well, yeah, I think that's maybe.
I think that's great, and sorry that I just forgot to introduce you. Yeah, you're, as you said, you're a PhD student in the Wageningen University in 2 groups, in animal breeding and genomics. Yeah, animal breeding and genomic groups and quantitative veterinary epidemiology groups. That's it. Am I right?
Yeah.
So I looked at your presentation, which was very nice. So I can start with some questions before other people put their question in the chat. Or I was wondering what about so some robustness analysis. So which is the dependence of your results on the underlying modeling assumptions? For instance, you assume that the individual susceptibility was dispersed, such as a log normal distribution, for instance. Yeah, this is one point. And you cited, I think your reference was a study by Lloyd and colleagues that showed that there were super spreaders in some diseases. So, well, so this was one question about robustness, but another related question is, did you perform a sensitivity analysis with respect to epidemic parameters? And are your results robust with respect to this?
Yeah, so an important, well, when I start with the distribution, the problem was that we incorporated susceptibility through scaling of the transmission rate parameter. So the beta is multiplied by the susceptibility of the individual. And then, of course, it can't be negative, because the transmission rate cannot be negative. And all the thing is that for convenience, it would be nice if we have a distribution with a mean of approximately 1, because then it's easy to scale the model through beta, so through the mean transmission rate parameter. What we generally do in animal breeding is assume that such traits are normally distributed, but of course we cannot do that really with this susceptibility, because then the chance to get negative values is always there. And then it sort of turned out that the log normal distribution was more or less the best fit.
It was really a fit, or because you did not
no,
consider?
I mean, the most natural thing to do it. But,
okay.
But there were some problems when we needed to increase the variance to very high levels, because then it becomes very much skewed to the right, of course. So we ended up with some very susceptible animals. So when the variance is not that high, this log normal distribution works fine, and then it's also, of course, it looks very much like a normal distribution. But with higher variance, it became more of a problem. Also because the mean then is more different from 1.
And so did you try other so for this distribution, but also for model parameters, did you try other values to see if everything is is kept?
Yeah, so it's mainly sensitive to the prevalence. So if we would have a very low or a very high prevalence, then it becomes then of course there's very little variation in the disease status of individuals. So what we then have is that only a few get infected, or almost all of them are infected the whole time. So then it becomes very difficult to find any heritability at all. So to detect any genetic variation. So that's in fact the main thing.
The main effect of
yeah.
There are additional questions. So Colin Birch would be interested to see your study on the potential of selecting for disease resistance. Are you aware of the initiative to provide access to TB resistance information on cattle in the UK?
No, I wasn't aware of that. No.
Okay.
No.
Maybe you can ask the question. Yeah, sorry.
Yes, can you hear me?
Yes, that's perfect now.
Yes, I'm afraid I haven't been able to check the actual website and so on, but yeah, there's an initiative that's part of some of the farmers obviously can obtain semen from a site that provides information about the genetic characteristics of the bulls, and one of those characteristics that information is provided about is resistance against bovine tuberculosis. So you might be interested in that.
Yeah, I will have a look at it.
Thank you for this comment. Pauline, you want to ask your question?
Yeah, you could have done it. I was wondering how you deal with the fade out that occur in your model, because once it has faded out, you do not reintroduce the disease. So how do you account for that in assessing the effect of selection?
Yeah, so you mean that when we selected the animals, then at a certain point the disease died out? Yeah. So that was just then the story ended, so to say. But that was already quite an interesting finding that was possible, because that's very much different from what the current ideas are on that. but we didn't estimate any. So I think I don't fully understand what you mean by assessing the effect of selection.
I mean, you were looking at, so you select for more resistant animals and you look at the impact on the disease spread, but finally your model is stochastic, if I remember well. And so you could also have just random extinctions that could hidden the effect of the selection. Or
well, yeah,
increase the effect. But,
yeah, so actually I did two simulations. So the first one without selection of animals, and the second one where we select the animals. So in the first one, I just started because we had endemic diseases. I started with a high enough prevalence so that the chance of random dying out was very small. And actually, I think that didn't happen in any of the cases.
Okay.
So we tried you tried to adjust or to have a control case in a certain sense, if I will understand.
Yeah. So we started without any selection to just find
just
out how many. Yeah. So to tune the model as well. Yeah.
Okay. Thank you. And Andrea has a question, and maybe I can also ask the last I would have, or almost last. So Andrea, I will give you the floor if you want. But so mine was, do you or other teams have plans to conduct in vivo experiments to test your theoretical results? And more generally, are we far from using this type of selection in the field? And so I think Andrea's question is more or less related. So maybe if you want to ask it, Andrea, or
Yeah. So I think Andrea's also her group is also very much working on this, and even more on the practical side, I think.
Yeah, no, it's very interesting because it's actually ground shaking. Because my impression is that the role of genetics for disease eradication is really vastly underestimated, and your study is a very important contribution in my mind to demonstrate that it is potentially underestimated. And as we all know, simulation studies are always very useful to set the foundations to say, look, it could be different as we observe. The question is really how to go from there to, because, should we look, is that the route to go to specific diseases? Is it better, who should we convince? Is it better to convince the genetics, the breeding community to say you should put more emphasis on disease traits, even if your heritabilities are low? Or is it more important to put the message out to the veterinarians or the epidemiologists to say you should consider genetics as another route for disease eradication? And just more, I wanted to hear your vision for that and what would be the next step. And Margaret may want to come on with that as well, I guess.
Yeah, so I think the breeding companies are really, of course, very important in this, because they, especially when we consider poultry or pigs, they are, or aquaculture, they are, I think, more leading than veterinarians in changing the genetics of animals. So for cattle, it's maybe a little bit different. So I think there is mainly, if we could convince them, and then, well, it would of course be nice if they do some selection experiments or with this, and then see whether it works in practice or not. That will be a great step, I think.
So
thank you.
Them to do an experiment to really prove it with real data.
Yeah, I think so. I think in practical conditions, it's really difficult to prove this.
Yeah, it was my question, because I'm not really from the field, but I was just wondering how could we prove it or just support
it?
it?
The problem is that the interactions between animals are very important. So that of course, if you have one animal with low susceptibility in a herd, that doesn't do so much. So they need to all have a genetic low susceptibility, or not, maybe not all, but at least a large proportion of them. So, and given that there are some problems with lowering production, etc.
Thank you. Maybe the last comment by Mart, and then we'll go. We need to move on to the last.
Yes, so I mean, there are of course many questions, and so one of the things is that that Peter Dries and myself are writing a theoretical, so that we derive this in general. That it and Andrea already has seen the presentation of Peter about that, I know. And so our strategy now is to estimate the breeding values for an endemic disease, and then so for a specific endemic disease, to automate the phenotyping and try to estimate really the breeding value for a specific endemic disease, which is very important in cattle. And of course, TB could be one of them, but we have picked digital dermatitis. And and then you should try to convince to breed against it the breeding companies. So that's our strategy. Whether
Your
that will
strategy.
work, I mean, I'm curious to see, but yeah.
Okay. Thank you very much for this enlightening discussion. And then we can, of course, to keep your camera on. So we will move to the last discussion around the presentation by Dibo Chakraborty, who is a postdoc at INRAE in the veterinary school, National Veterinary School in Toulouse. And your presentation was about phylogenetic investigation of control measures during a highly pathogenic avian influenza A, H5N8 in France in 2016 and 17. So please, maybe you can just summarize in 1 or 2 minutes your work, and then we can discuss. Thank you.
Thank you very much. Thank you for giving me this opportunity to present the work. I really appreciate it. So as you said correctly, that I studied this H5N8 avian influenza, which I think all of us know that it's a highly pathogenic avian influenza. So it's a disease of the poultry and of the wild birds. And I specifically studied these outbreaks during 2016-17 in France. So this was a part of a bigger epidemic in France at that, I mean, in Europe at that point of time. And France was particularly badly hit. I focused on only the southwestern part, which has the main part of it, the outbreaks. And what we find, so the question that we asked, that we focused on mainly two measures. One is the culling measures, which happened in 3 phases. 2017, 4th of January, 14th of February, and finally 21st February. And then also there was a ban on movement between different types of poultry farms, mainly from breeding farm to foie gras farm, and from foie gras farm to essentially just if there is an infection in a farm, any movement was stopped of the birds. And we essentially wanted to see if these have any effect. For phylogenomics methods, there are broadly 2 things people look at. Number 1 is the viral dispersal history, how the virus is moved. And more recently, actually by the first speaker, Simon Delacour, they have used a method of looking at measures using some kind of statistical tests, if certain kinds of control measures and their effect can be measured. We kind of use the similar philosophy, but in a very different model. The model that we used is recently developed. It's called a mascot, which is essentially a structured coalescent model, which in turn is like a population genetics model, which divide the whole populations, like whole outbreaks into different populations, and try to understand movement of viral lineages between these subpopulations. And then it also uses a GLM framework, where it tries to understand using multiple co-predictors, such as predictors associated with the control measures, also other kind of agricultural predictors, to understand if any of those had any effect. Broadly, the take-home message that we find is that the first culling measure that was implemented in a large region on 4th of January, that seemed to have the most prominent impact on the viral spread. Or more, in other words, that it seems that the viral spread between different departments, which is a particular administrative mid-level, I suppose, administrative division in France, there was reduction in viral spread between these units. Second, which we found, which is a weak result, we found that viral movement, the truck movement or the bird movement within departments were weakly connected to viral spread. The effective population size, which kind of tells also about how virus spread between farms. But between departments or long distance departments, if the departments which are far apart from each other, they were not associated with viral spread, which essentially suggests that possibly long distance truck movement or bird movements were not associated with viral spread, maybe some weak evidence of within department. We also found that the case count number, that is number of infected farms, is a very good predictor of effective population size, which is kind of a proxy for epidemic size. So that's it in a nutshell, the whole study is about.
Thank you very much for this quite exhaustive summary. Sorry. I looked at your presentation. It was very interesting, and I was wondering. So, what? So, so I tried to formulate my question, but it was
Sure.
which were your main results that could not have been obtained if genomic data were not used, or if you use only epidemiological data. So, well, yeah. What is the main, the main thing you found that could not have been found without this data, this genomic analysis, phylogenetic analysis?
That's a very interesting question, actually. And I think that's a very important crux of the usefulness of this kind of data. I think the results that we find are not okay, people have already used only epidemiological data to model these epidemics, and whatever they have found is very much similar to whatever the overall results that we find, that is that the culling, so they did not look at the large scale culling, for example, they looked at small local scale culling. So that is, I think, one of the new or kind of novel result that we find from our results, that looking at large scale culling, how that might have affected the movement of the virus.
But did this come from the use of genetic data, or it was because you thought about including this potential predictor in your model? I'm not sure.
So yes, not really only. So the predictor that we could use, that is because of we have genetic data of that. So the model that we use is a population genetics model, so which is connected to the data, right? And in the population genetic model, we can actually think about how the virus is moving between populations, which because, and correct me if I'm wrong, because I'm not an expert on epidemiological modeling, but if you only have epidemiological data, which is like a case count data or which is observed, is possibly not, so you only see how how epidemiological patterns is happening, while the genetic data is telling you how the virus is actually moving. So I think using this kind of models which uses genetic data, you can actually be much more you have much more insights into how the virus is actually moving instead of how only the host is moving, which you find.
It's you can disentangle, maybe disentangle. Sorry. Well, yeah. So a point of detail. You said, if I were well, I remember in your presentation that the density of duck farms it was not included in the final model for the prediction of the efficient population size, and you said that it was in contrast to previous studies not including genomic data. So this could be another result that would not have been obtained without genomic data. But how can you interpret this? Is it a bias, or is it on the contrary, is a very interesting finding?
It is an interesting finding. So as I said, that when we looked at this effective population size, we find a weak effect or weak result suggesting movement, that live duck movement. But then we also looked at all the different co-predictors, right? And the live duck movement actually is collinear with the duck movement. Sorry, the duck density, the density of the duck farms.
Mm-hmm.
So because these models, the GLMs, the way they are designed, it takes care of all of these different co-predictors and their effects independently, or at least they are supposed to be kind of an average method. So what we think is that the movement of birds within departments, they are an outcome of multiple factors, and one of them is duck density of duck farms. So I would not like to really overinterpret the data, but it is possible that there are some aspects of the movement that is addition to the duck density that is possibly is getting captured in this result. While in earlier studies, which did not really looked at, I'm talking about genetic studies, which did not really differentiate between different kinds of movement or the different components of a movement which is associated with duck density, or just the number of trucks, a number of movements independent of duck density. So there can be some possibility there, but I think it requires much more investigation. At this point, really, we are not sure if this weak result that we find associated with the movement of movement of the live duck trucks are independent of this density of duck farms.
Okay. And maybe there is a connected question by Pauline concerning the effect. Yeah, Pauline, can ask it.
Hi, Devo.
Hi,
I was wondering, because some of your effects that were not significant were a bit, there was a tendency, and I was wondering, I'm really not aware about how many data are needed to do such analysis, but maybe it could be an issue of statistical power. So maybe you have not enough information to see the effect. So I was wondering how you can distinguish between an absence of effect or a lack of power. Or maybe it's a bit naive question, but
no, not at all. I think it's a very valid question and very important question about these kind of methods. Okay, I think there are two questions in this one question. So first of all, the effect size that we see is not actually the effect size of the co-predictor. I just wanted to clarify that. This is because of the method that BSS-VS that we use, which essentially the effect size, it essentially means that the co-predictor, if it is correlated or inversely correlated with the viral spread or any. So it's not the effect size of the predictor. So what essentially this result tells us is that if that particular co-predictor is positive, that means it's correlated with the viral spread. And because, for example, the 4th January, the way we coded the predictor is that before 4th January, we assumed that there was viral spread. And after 4th January, we assumed that there was no viral spread. And because it was positively correlated, that means this predictor was significant. That means we concluded from there that viral spreads reduced after that particular 4th of January. So it's not really how much effect was there of this culling. I just wanted to clarify that part. Second part is the way the BSS, so the second part I want to explain now is, which is more directly of your question, is that the effect of the data size is there. I would suspect that definitely the data size would have some effect, but this method has been developed a couple of years ago and 2 papers have been published and they have shown simulation studies using this method across different size of data, and what they show that the data size is quite robust. So it's essentially the results are not very much influenced by the data size. But I would expect that there has to be a limit to that. And from our results, what we find is that using these kind of a model average, BSS-VS is kind of a, again, I'm not an expert on Bayesian statistics, but whatever I have read, whatever I've understood, is kind of a model average method, and it actually takes into account all these different parameters with, and if the data were not sufficient, we would not possibly have found any results or any significant results.
Okay.
We did take care of, because we ran MCMC, so we did take care of all the equilibrium state, and we take care of all the bonding, everything, all those things.
In the Bayesian
So-
paradigm, if you don't have enough data, you will find in the posterior what you put in the prior. So more or less.
Yeah.
Right.
Yeah.
Thank you. Thank you very much for this, for answering our questions. And sorry, because since we are on time, perfectly on time, but it's a bit late, so we are all want to.
Yes. Thank you very much.
Thank you very much, Zibo.
Giving me the platform. Thank you.
And I would like to thank all the speakers for the very interesting presentations and discussions that follow the presentation.